Hello, everyone, and welcome to another episode of Emerge. My name is Stoyan Stoichev, and I'm part of the Resin Biosciences team. Today, I have the pleasure of hosting Dr. Seamus Morone, who is currently based since actually 2019 at the Institute of System Biology in the lab of Rob Moritz. Where he largely focuses on mass spectrometry to profile a human phosphoproteome. In terms of his background, Seamus has a lot of experience in structural biology, having done his PhD in Johns Hopkins University, where he focused on utilizing cryo-electron microscopy to study proteins from the innate immune system. And subsequently also did a postdoc in the Duke University of Singapore, where he also used cryo-EM to study dengue and Zika viruses. But today he's going to focus on some of this work where he systematically characterized the different data acquisition parameters, particularly on Orbitrap systems, but also with different front ends, LCs, and different phosphopeptide enrichment chemistries in order to get the optimal coverage and localization confidence from phosphopeptide enriched samples. So, Seamus, thank you very much for taking the time to do this webinar with me. I really look forward to your presentation.
Thank you, Stoyan, and thank you for the invitation to present my work. And can everyone see the screen all right?
Yes, looks great.
Okay, great. Okay, so today I'm going to talk about the work that we've been doing on phosphoproteomics in Rob Moritz's lab, specifically using an Orbitrap instrument in DDA mode. And we're exploring some of the effects of the parameters on the quantity and quality of phosphopeptide identification. So I'm sure we're all aware of the importance of protein phosphorylation. It's one of the most ubiquitous and arguably one of the most important post-translational modifications that proteins undergo. And it has a diverse array of roles in all domains of life. It can act as molecular signals, switches, timers, structural components. And this is underlined by the fact that one of the central processes of cells, the cell division pathway, is critically regulated by phosphorylation and dephosphorylation of the cyclin-dependent kinases, which in turn, as they're kinases, they are phosphorylating other proteins. And a study came out now about 17 years ago that characterized the human phosphoproteome and estimated about 75% of the human proteome may be phosphorylated. But it's important to note that this doesn't necessarily mean phosphorylated at any one time or place, as one of the key characteristics of phosphorylation is its dynamic nature. Besides its labile bond, it changes within short frame time frames and in distances as well. So a cell in one tissue may be expressing a certain subset of phosphoproteins, whereas the cell in a different tissue will be expressing perhaps a completely different set. And even within that tissue, over time, this will change. So 75% over the course of time and place. And because of its central role, you can imagine that dysfunction or aberrant phosphorylation plays roles in many diseases, including multiple forms of cancer. So not only is it of interest for basic science, but also for drug discovery and therapeutics. Arguably, the gold standard for phosphoprotein identification is mass spectrometry, which in turn is a vast field. There's many different modes of acquisition, and I will be focusing exclusively in this talk on data-dependent acquisition, and specifically on the Orbitrap Lumos using electrospray ionization with HCD as the fragmentation mode. But yeah, there's a vast array of different mass spectrometry acquisitions. And one of the reasons why it's such a gold standard is it provides good sensitivity, can cover several orders of magnitude of dynamic range of the peptide abundances, has very good throughput, it's capable of analyzing many samples per day, which is an ongoing field of research. And you can multiplex by combining multiple samples via labeling into one run. And it allows identification and localization just in raw data without any hypothesis to drive the acquisition of the data. And it's also fairly well worked out in terms of sample processing. You start with a source of tissues or cells, you extract the protein, digest them, perform enrichment, and fractionation is also important, and then you run them on the instrument. So mass spectrometry is a pretty well developed field for phosphoproteomics, although it is very active. And phosphoproteomics itself presents many challenges that are also useful for mass spectrometry to tackle. So as I mentioned previously, it's very dynamic, and samples even processed at one time point may be different from samples processed at a different time point, which requires a very good sample processing technique. It requires moderate amounts of starting material, although I will touch on this near the end of the talk, as this is also actively going on right now with reducing the amounts of starting material. And it requires additional processing steps than a typical cell lysate preparation. And what I'm referring to here is enrichment, which is, as I will show, is a necessary step. So tackling each one of these points in a little more detail. So phosphorylation is dynamic, and samples processed. It's critical for having a good time frame, because there's these studies that have shown, for instance, this blot against total AKT versus a phosphoform of AKT. And what the researchers did was just excise tissue, allow it to sit for a certain amount of time, and then finish the processing. And what they showed was that total AKT showed a steady, pretty linear decrease over time, whereas the phosphoform showed this drastic exponential decrease, so that even after 30 minutes, the majority of this phosphoform was already gone from analysis. And then another study has shown that even within a shorter time frame, 20 minute stimulation time frame, even within a single protein, multiple phosphosites undergo complex and heterogeneous dynamics. And even within a pathway, multiple proteins shows the similar complicated behavior. So that this emphasizes that parallel processing would be important for good replicability. So in this case, we're talking about automation and high throughput, which have also been discussed in previous merge episodes. So the second point, it requires moderate amounts of starting material. So the field's progressing, especially with the standards triptych digests, towards less and less samples. So even with 25 nanograms of starting material, can characterize and quantify over 3,000 proteins. Conversely, with a phospho-enriched sample, 1,000-fold more material, 25 micrograms, yields about 3,000 peptides, so considerably less than 3,000 proteins. So this emphasizes that more material is useful for phosphoanalysis, and this is critical when you are working with precious samples. And then it requires additional processing steps because there's a big difference between unenriched and enriched samples. So here in our lab, we've run an unenriched sample of halolysate, and then we get a certain amount of spectra, peptides, and proteins from this. If we focus on just the phosphoforms within the sample, we find only less than 0.5% of spectra and peptides that are phosphorylated. And so starting with roughly the same amount of material injected on the instrument with an enriched sample, so this is after phospho-enrichment, we're getting similar amounts of spectra, but fewer peptides and proteins, and this is reflected in the less complex chromatogram. But conversely, we're getting a huge boost in the number of enriched spectra of phosphopeptides and proteins which contain phosphopeptides. And so this just emphasizes that enrichment is a necessary step. So a pipeline for phosphopeptide processing and analysis, as just mentioned, is divided into two parts in our processing. So the first steps would need robust, reproducible protocol so that we're not getting huge variations in our sample processing. And so this is also tied to that less steps and less manual intervention is ideal, as this reduces some of the stochasticity, and faster processing time, as we have just explored, is important to limit the degradation of the phosphopeptide samples. On the analysis side, what we need is strong sensitivity, as many of these phosphopeptides are in low abundance, good identification rates so that we have a large number of phosphopeptides to work with for statistical analysis. And generally, we want high confidence in the localization, although this will depend on the nature of your experiment. And I just want to sidestep briefly to talk about unlocalized versus high confidence localization, because identifying a phosphopeptide can be as simple as just, does the precursor have this mass difference shift of a phosphate group of 80 Daltons? But identifying a specific site requires that the fragment ions at where you believe the phosphorylation is have this shift. And it's complicated by the fact that in biology, phosphosites often occur near other phosphosites, and multiple phosphorylation is common. And often these sites are differentially phosphorylated. So it's one may be phosphorylating, the other not, and that may represent one part of the pathway, whereas the inverse may be a different part of the pathway, and sometimes both may be phosphorylated. So you need evidence for phosphorylation at the residue, as well as evidence against it being elsewhere, or at least stronger for it being at one. So in this case, if we've got two phosphosites, we want to weigh evidence for it being at one phosphosite or another, and we can see that there's more evidence in support of it being at one over the other. And this is mainly due to the spectral quality. So improving the spectral quality will improve the localization. And in the field, there's the standard of the class 1 site that I will be using. And this is a localization probability at the site of at least 0.75. On the analysis side, we use the transproteomic pipeline, which it's a suite of tools that allows us to go from the file provided by the instrument, such as a raw file from Thermo, to a final probability-based table of peptides or proteins. And the suite's been developed at ISB with software that's free and open source, developed both at ISB as well as other labs that we've collaborated with. And it's being constantly updated. The new version 6.3 is coming out shortly. And there's a large community participation. We have online discussion boards that allow people to post questions or insights. And this allows the authors of the software to respond with their own help, or more experienced users to jump in and help out. And for the pipeline that I have been using for these results that I'll be showing, I'm usually stopping at the PTM profit level, which is peptide validation for post-translational modifications. But one can go on to validate at the protein level to get a better FDR for the protein level. So tackling how we have investigated these the phosphoproteome, we started with the processing methods using a semi-automated technique. So previous talks in this emerge have mentioned or focused on the protein aggregation capture, which we have adapted in our own lab for phosphoprotein processing. And just briefly, as it's already been discussed in detail, but you start with soluble proteins that you've solubilized in a cheotropic agent. You add organic solvent to precipitate them, and they form aggregates. And then you add microparticles, and the aggregates absorb onto the surface. And then using magnetic beads and a magnetic liquid handling robot, you can move these from the cheotropic buffer into to buffer exchange it into a buffer suitable for digestion. And then and proceed with the enrichment steps after this. And a paper came out a few years ago showing that this PAC method is an improvement in terms of phosphopeptide recovery over the standard in solution method. And there have been several studies that have used the PAC method in combination with the phospho-enrichment method, which the Luchard et al. Group have referred to as R2P2, which we have adopted for our own uses. And so after digesting the proteins, we proceed with the enrichment. And we have been using a combination of titanium and zirconium IMAC beads provided by resin for our enrichments. Mainly this combination is because a study came out just 2 years ago showing that zirconium and titanium capture slightly different distributions of phosphopeptides. And so in order to get a larger range of chemistries of our peptides, we combine both of them. And we have tested this combination both with the standard IMAC beads as well as the next generation. They have released the high performance beads, and we have seen a boost in both the number of raw phosphopeptides, but a much larger boost in terms of the high confidence localized peptides using these high performance beads, which we would recommend. And then we have been using the Kingfisher / BioSprint instrument for this entire processing. So on the analysis side, as I mentioned, it needs a few details, and we need to optimize several parameters because if you're just using a standard standard method from that Thermo provides when you begin an instrument method, you have there's a standard settings that you can optimize for whole tryptic digest of cells. And then if you consider a phospho optimized method, which many published studies use certain values for their method, and you contrast these two from an unenriched sample, you're getting similar return on peptides. But if you look at a phospho-enriched sample, you can see that there's a large difference from a standard whole cell lysate optimized protocol versus a phospho method. There's a boost in terms of raw phosphopeptides, but there's a much larger boost in terms of the high confidence localization. So the question is, what role are the parameters playing between these two methods to provide this boost in phosphopeptide identification? Because there are several that you can optimize on. At the MS1 and MS2 level, you have maximum injection time and automatic gain control. You can optimize the collision energy, the number of precursors selected for fragmentation, At the LC side, there's gradient length, and then there's plenty of others, such as isolation window, that you can work with. So one of the take-home messages of what I'm going to present, or what I have been presenting, is that it really depends on what one is after. So you want high sample throughput, then you want to focus on short gradients, etc. You want high phosphopeptide numbers, or high confidence in those phosphopeptide localizations. All of these will play a role in how you optimize your parameters. And there have been lots of good studies that have investigated many of these parameters on a lot of them on whole cell lysates, exploring what role they play. And then a few have also investigated it on the phosphopeptide level. Particularly, they've been focusing on collision type used for fragmentation. But we really wanted to explore what AGC, IT, and several other parameters that we looked at, which I'm not going to touch on here time reasons, but you can always look at our paper, which we put up into Chem Archives. So just as a refresher, automatic gain control is just a measure of the ion population in the C-trap that accumulates and then sent for analysis to the Orbitrap. And you can increase or decrease the amount of ions by controlling this value, but conversely, you can also accumulate more chemical noise as well as other dangers. So it's important to choose a good value for this. Maximum injection time is just the time spent accumulating these ions, and can lead to long duty cycles as you increase this. It's important to note that they're complementary. So the instrument will look at both set values, and whichever is reached first triggers the end of the scan. So if you set a very low AGC and a very high IT, chances are it will never reach that maximum IT. It will always be triggering on the low AGC. So yeah, in what I'm going to present, I'll just be showing the MS1 and MS2 level of IT and AGC and its effect on phosphopeptide identification and localization. So the first one we explored was injection time, and as we varied it from 2.5 milliseconds to 40 milliseconds at the MS1 level, we just saw a little effect on the total cycle time. So we can see that as we go from low to high, there's a slow decrease in the ones that actually reach this maximum injection time. As we accumulate more ions, it begins to trigger the AGC instead of the maximum injection time, even with the AGC set at its maximum value. And this is also reflected in the duty cycle and the scans per second. We lose about 1 scan per second on average as we increase the injection time. So that's about just a 20% fewer scans compared to the lowest acquisition time, or the lowest injection time. And then if we look at the raw number of phosphopeptides, we see a slight drop in raw numbers at the very high end of injection time. And this corresponds roughly, it's about a 15% drop, roughly to the effect that we saw in cycle time. So this is mainly due to an increasing cycle time, not allowing a number or less identifications. And we saw no effect essentially on the localization confidence. And we weren't expecting to see any effect on the localization, as this is just MS1 IT. So all we're doing is accumulating precursors for subsequent selection for fragmentation. But what we do see is a shift in the population of intensities from high precursor intensity at the low injection time to a lower distribution at the higher injection time. So as we increase the injection time, we're starting to pick up more low intensity precursors, less abundant ones that can be chosen for subsequent fragmentation. So So turning to MS2 injection time, it's a slightly more complex story. So as we change from 15 milliseconds to 240 milliseconds, there's kind of conflicting behavior between the raw number of unique phosphopeptides and the fraction that are confidently localized. So we see there's a peak in the total numbers of between 60 and 120, and then a drop off at either end. And then with the localized fractions, we can see a steady increase as we go from low to high. So this is partly due to more ion accumulation, which allows for a higher quality spectra and allows better localization. So as we go to these long time points, we're getting much better localization. But also in conflict with this is that we're increasing our duty cycle, of course, as we go from a lower to a higher injection time. And this is reflected in going from about a 1.4 second cycle time to 3 to 4 times higher at the highest injection time used here. And if we look at MS1 AGC, we see also some conflicting behaviors. So as we increase it, and each one of these steps increasing by tenfold, we're seeing this plateauing of the unlocalized phosphopeptides, but a decrease and leveling out of the fraction that are confidently localized. And so as we go to the higher AGC, we can see that there's once again this shift from the average intensity being at the high end to a more broader distribution towards the lower end. So at this low AGC, we're getting few ions that are being fragmented that are generally the most abundant precursors with high ion population. And as we increase this, we're starting to pick up less abundant precursors with lower signal. And this is reflected in the fraction confidently localized as these lower abundant ones have a less strong MS2 fragmentation signal. And if we look at the effects of MS2 AGC on phosphopeptides, we can see that as we go from low to high, we see fairly low confidence at the low values, and then an increase in leveling off at the high values. And with the with the total peptides, phosphopeptides, we see a peaking and then a decline and leveling off. And this leveling off, however, is most likely an artifact of the fact that we are never really accumulating this highest AGC value. As we can see, the number of scans that are actually less than the maximum injection time after about 1E4 are never reaching the majority. So instead of being triggered at the high AGC, we're being triggered by IT, which is unfortunately unavoidable as we're using the maximum settings for IT here. And as we can see, this cycle time, we set the maximum for 1 second. So anything higher than 1 second, we're already at this point reaching 14-second cycle time with very few identifications, starting from a 1.4 seconds. So it's an order of magnitude larger by going up to these high values, which makes it untenable to use such a long cycle time. But what we do see from this is that with fewer, at the lower end, we're accumulating fewer ions, which leads to these low identifications and fairly low confidence. And then as we increase to a higher AGC, we're getting higher ion populations in the fragmentation spectra. And but unfortunately, we're getting a longer duty cycle, which leads to this lower amount of identified phosphopeptides, but they're higher quality spectra, so it leads to high localization confidence. So we can kind of look at these as a table, and as I mentioned, the take-home messages of this is that there's trade-offs in choosing these values. So usually at low values, you have certain advantages and disadvantages for the AGC and IT. And then these at high values generally switch around, but sometimes the behavior is a little more complex. So it's important to choose values based on what you are looking for. As I mentioned, you want fast processing, precise localization, large numbers. This will involve optimizing and probably having trade-offs. So some of these parameters conflict with each other. So if you want to maximize one, it could be to the detriment of the others. So if you want to optimize for localization, that generally involves slowing the instrument down a little bit to accumulate more ions, which generally translates into fewer identifications. So you can't have both worlds, unfortunately, with these settings. And there are other parameters, as I mentioned, we explore several in the paper that come with their own trade-offs and are equally important. And I didn't get a chance really to talk about the LC portion, but yeah, we explore also the LC side of it in the paper. So yeah, a number of these points I have mentioned are actively ongoing research right now. So shrinking sample size is becoming a important direction people are going in. A paper just came out last month, I think, or a month before, Microphos, where they're working with 20 micrograms of material and they're getting over 10,000 confidently localized phosphopeptides with their setup. And then there's previous emerged talks that have talked about shrinking samples and getting equally good results. So the field is progressing more towards shrinking the samples. Also, localization numbers being closer to the total phosphopeptides identified by tweaking these instrument parameters or using the next generation instruments is becoming more important. And then fuller automation. There's previous emerged talks that have talked about fully automating everything. As we're using, I would say, a semi-automated method with the Kingfisher, there's still more room for removing the human component to allow for more robust and reproducibility. And then there's also stronger papers that have been coming out showing how DIA has been improving over the DDA results for phosphoprotein identification and localization. And DIA passive is one of the methods that is being used to be comparable to DDA, and with the advantage of shrinking the length of time spent acquiring the data. And then besides serine, threonine, and tyrosine, there are many other amino acids that undergo phosphorylation, histidine being one of them that has extensive research about it. But there are several other amino acids that people are investigating and looking for diagnostic ions that will help them identify and characterize these. And then probably one of the most important is annotation. A paper has recently said that, we have several phosphosites within the human proteome that we have characterized by mass spec or other means. But only 5% of these actually have some sort of biological role that we know of. So it's important for us to try to get out and figure out what these sites actually, what role they play. So I just want to thank Rob Moritz and everyone in the Moritz lab that has contributed to the studies, several of them within the lab, and then Stoyan at Resin for the opportunity to present this data, and then all of you for tuning in and listening. Thank you.
Thank you, Seamus. Thanks for the informative talk. I think it's, always great to see the balance between all these different parameters to get the optimal identification rates. And I suppose maybe the first questions I wanted to put to you is, what about quantification? Is there balance there? If you want to also apply some kind of quantification in the DDA, probably on precursor level, how does that look in terms of the various parameters you've tested?
Yeah, so I quantify in the TPP using Express, so it's a label-free method. So yeah, it's the label-free method. Sometimes you have to really dig through the data to make sure that it's quantifying correctly. But yeah, I usually am quantifying to have 5 points per peak in the MS1. And yeah, I do see similar shifts in the peak areas, as I saw with the shifts I showed in the presentations. So that as you go from low AGC to high AGC, you see a shift in the peak areas from mainly high to a more wider distribution. You start seeing more low abundance peptides. But yeah, I mean, one of the challenges is actually figuring out the occupancy of these sites. But unfortunately, with the method I was using, we can't quantify the occupancy to any believable extent as we enriched on it without having a dephosphorylated fraction.
Okay, and I suppose related to this, it's something you maybe didn't cover, but you touched on is gradient when the front end LC. With the current setup, what have you found to be kind of optimal, the optimal range?
Yeah, so we were focusing on the top precursor selection, and yeah, unfortunately I didn't include a slide, but as we went from 5 all the way up to 30, we saw this kind of plateauing behavior, and it peaked around 15 to 20 on the Lumos. And when we explored on the Eclipse, I believe it was at 20 that it peaked. And then we also see a shift in the number of identifications from going from a 60-minute gradient and extending that gradient of 5 to 35% acetonitrile from 60 to 120 to 240. We see after 120 that there's really no difference in the number of phosphopeptides, but at the shorter gradients, we're getting a higher confidence in localization. So that once again is kind of playing a role. And I didn't quite emphasize it in the talk, but confidence in localization, at least as a percentage, may not be too worrisome to you if you're getting 20,000 phosphopeptides and 80% confident localized. It's far better than getting 3,000 at 90%. So yeah, but yeah, so shorter gradient led to higher confidence, but lesser identified.
Yeah, and this is probably the effect of DDA and cycle time and not be able to fragment everything.
Yes.
So moving maybe one step forward to the pipeline in terms of the phosphopeptide enrichment, what do you find is the major take-home messages? I know you've tried different combinations of chemistries. Do you see a gain of doing combinations of various IMAX in series or in parallel, or is it still best to still use one chemistry and there's a lot of losses once you start putting the samples because of ion separation?
Yeah, I mean, I think a lot of papers have shown that you do get different distributions of peptides with using different chemistries, particularly like using titanium dioxide and IMAC in combination, which unfortunately we haven't really explored. We've mainly been focusing on IMAC beads. But yeah, I mean, I would think that using a wider, diverse set of beads might be better. Although, yeah, you may need to use several samples and combine them.
Yes, and it's always the balance between, if you're going to doing enrichment from each of these different chemistries, do you do them in parallel? And if you do, then you have recent mass spec time. At least in our hands, this is kind of the chain where we will see quite an increase if you do, metal oxide versus IMAC or different types of IMACs. But once you pull the samples into one, then those gains are quite reduced. But something we've observed is, and also that's been now reported, is that doing multiple cycles on the same sample, you can actually gain, especially with low input material, we can gain quite a lot of identification. I'm not sure if this is something you've done. I think you do multiple cycles of the same IMAC beads. Yeah.
Yes, yeah. So we would do 2 rounds of enrichment on the same sample, and we did find a boost. Although the second round tended to have more carryover of non-phosphopeptides, but yeah, we found that still it was offset by the total number of confidently localized phosphopeptides that we got. So we figured the trade-off was worth it in that case.
Yes, I think so.
Yeah, and using the combination of titanium and zirconium, we found we did have to optimize a little bit of the buffers, since I think the recommendation was 0.1 molar glycolic acid for zirconium. Recently, I think it has now been lower for titanium, but it seems titanium a little bit higher is better.
That's correct, and it's almost like a fine-tuning key that's, in terms of selectivity. So zirconium, we also prefer to use it at 0.1 molar. I think multiple cycles of zirconium IMAC enrichment and maybe reducing the bind time for the second cycle to reduce the non-specific interactions could be one way to go. I think.
Yeah, that's interesting. That would be worth checking out. Yeah, because we generally use the same binding time for both rounds. Might be worth it to explore that.
Yeah, that's something that's we've seen that could work. Let's see. I don't see any more questions. Maybe one from my side about bottlenecks in this full pipeline. What do you the method is kind of semi-automated and going to the LC, and it seemed like you have a kind of pretty automated pipeline for data processing. But what do you see the major bottlenecks, and where do you see the major advancements to increasing the throughput? We talked about DI on the mass spec side. We can probably do shorter gradients. On the front end, is it the desalting that is basically taking the most time, or is it maybe the extraction is causing the most bottlenecks?
I would say on our end, the bottleneck is the sample run times, which in our mode, I mean, we've been doing a 2-hour run, so and generally we have a blank in between or a standard in between, so we can process 6 to 8 samples per day or so. So increasing, that's probably the biggest bottleneck. Everything else, I mean, you can work, like for instance, you said the sample cleanup. We've been using rack tips, basically like a Evo tip or a Finicep tip, and you can use 96 at a time and spin those in a centrifuge or whatnot. And so that's not much of a bottleneck. But yeah, I would say the sample processing is less of a bottleneck than the actual mass spec time. So DIA may be the best way to go for reducing that runtime, which we're starting to explore.
Cool, and we look forward to that, to the data. So I don't have any more questions. I would like to thank everyone for joining us. We are going to post the recording online as well, so if you want to review anything, and I'm sure you can also contact Seamus directly if you have any more questions. And thank you for your time for a relatively early morning in Seattle. And see you guys. Yes. definitely. And see you guys for the next episode. Thank you, Seamus. Thank you, everyone.